and turn it over to Kim Buckley for introductions. Enjoy your class.
Good afternoon, everyone. My name is Kim Buckley, and I'm a member of the SMT committee and coordinator for this presentation. George Allheit is our presenter this afternoon. George retired from the Department of Physiology and the Institute for Neuroscience at the Northwestern University School of Medicine, where he was a research associate professor. Prior to Northwestern, he was a research professor in the Department of Psychiatric Medicine at the University of Virginia. Allheit earned his BS in psychology from the University of Illinois and his PhD in biopsychology from the University of Chicago. He earned postdoctoral fellowships in psychopharmacology in the Department of Psychiatry at Albert Einstein College of Medicine and in neuroanatomy at the University of Aarhus, Denmark. So without further ado, George, take it away.
Well, welcome everybody. I'm going to start my presentation on good advice to give you a little sense of the directions in the brain in case you haven't done it. It's a little confusing. It's sort of the vocabulary you need to find your way around the brain. If you've read the handouts, this will be a little redundant, so I won't try to drag it out. But these names in terms of directions are like the compass rose, so you know which way we're going. Anterior, posterior, dorsal, and ventral. Really, if you look at a vertebrate, just a little out of a line, in a fish, there's a dorsal fin, a ventral fin, rostral, and caudal. Caudal means tail, rostral means beak. And these names just filter into the names of things in the brain. And the alternative to rostral and caudal is anterior and posterior. So when I say this, that if you look anterior, you have to look toward the front of the brain, posterior toward the back, and usually I'll try to give you a hint on where we're going. This is a horizontal section through the human brain, but we'll also see horizontal sections in a rat brain. That's usually, in a human brain, it's usually between the eyes and the ears that they line it up, but there's no fixed horizontal plane. It's arbitrary, and even for people who do the medical imaging, haven't really decided on what to do. In the old days, the anatomist would look at this particular little dot down here called the anterior commissure, and there's one in the back, and they like to look at their histology and make the horizontal line go through those two little anatomical landmarks. This view, looking from the side, is called a sagittal plane, and this, of course, is looking at the left side of my face, or is it the right? I'm not sure anymore. But in the sagittal plane, runs straight down the middle or parallel to it. And that's the actual one real landmark that you have in the body, because there is a plane that divides us more or less symmetrically in half. This is what's called a coronal plane, cut perpendicular to the other two planes. And again, the angle is a little bit arbitrary. In this case, it's perpendicular to the horizontal and the sagittal plane. Once you've established two, you've established the rest of them. And then I've named some of the structures that we're going to be talking about. We're going to be talking about the striatum, which on my screen doesn't look as good, I hope as it does on yours. This is a large mass underlying the cortex, which is larger in lower animals. It's called a striatum because in tissue, you can see like little stripes of axons going through it, plus one big massive bundle. So the name is descriptive. Septum, descriptive, it divides right at the middle of the forebrain. Forebrain means the front and the base of the brain. And the bed nucleus of the stria terminalis we'll get to later, one of my favorite places. And I'll try to give you a better picture, but the globus pallidus is just inside the striatum and part of the larger complex that forms this little angle underneath the cortex. The amygdala is right underneath all of the basal ganglia, the striatum here in the globus pallidus. And at the most further front of the basal forebrain is something called the nucleus accumbens. This is just a reminder, in seahorses and in humans, the dorsal and ventral directions really are perpendicular to the neural axis. So on the spinal cord, which we'll not get to, but we'll get to the brainstem, normally anatomists will take dorsal and ventral as you go from the ventral, the belly, to the back. That means this angle changes as you go around this arc. It does it for seahorses, it does it for us. This is just a picture of a rat skull. In a rat, we arbitrarily set this up just level to the cortex for work. So the horizontal in a rat is a little bit better defined. We get to cortex. There's lots of little subdivisions that people publish, but the two main divisions that we'll talk about just for the sake of following what's going on are the sensory cortex, which really comes all the way around here up into right about here, which is where information comes into the brain from the body, from the auditory system, from the eyes. And then frontal cortex includes an area that's directly related to the muscle groups of the body, which is called motor cortex, and the other place, which is called prefrontal or premotor cortex, where most people think the good stuff happens, like executive decisions and things of that sort. This is part of sensory cortex. This is called the temporal lobe. It's because it's right over, it's right over the auditory canal and underneath the temporal bone. This is part of sensory cortex that extends here beneath the motor and frontal cortex. And here's, I put this arrow so you can keep track of where dorsal ventral orientation should be. And also when we're looking at here, you can see this line gives you an idea of where you are in the brain. So here's the brainstem, and the brainstem, because of the position of our head sitting at a right angle, this is an area called Substantia nigra. In the temporal lobe, right behind the amygdala, is a nucleus called the hippocampus, and the amygdala is right in front of it. If we look at the brainstem later, which we will, you see you have to turn this around because this is dorsal here and this is ventral. Whereas in a horizontal section, this is anterior and this is posterior. It's a little confusing for humans, but in rats, it's a lot simpler. So here's just another view of the striatum from the side, which I've tinted blue. Medial to it is the thalamus. Just like the cortex, the thalamus can be divided into a posterior part that gets sensory information and an anterior part that processes motor functions and more exotic things. Well, this is a dissection of a human brain, where you can get to see what this looks like. This is the striatum, and I've tinted it, excuse me, I've tinted it blue. So basically this part of the striatum sits along the lateral ventricle here, so it's free, and the surface is just the way it is. The other part, we scraped away all the overlying cortex, and this large band of fibers in a human is called the internal capsule, which connects the cortex and the brainstem and the thalamus. And then there's little stripes that go through between the connections that give us its name striatum. So here's a picture of the striatum again, the nucleus accumbens and the septum. That's redundant. But here's a little better picture of the striatum. This is immunostained for substance P, which is a peptide that's a neurotransmitter in the brain. So by using antibodies, we can visualize where this is in the human brain. And you can see the stripes, perhaps a little bit, maybe not, the stripes that go through in the internal capsule that divides this into a dorsal and a ventral part in humans and primates, but not in things like rats and dogs, like rats and mice. So this is the internal capsule. Because of the human anatomy, this is often called the caudate because it forms like this large arcing structure that looks like a head with a tail going back. That was the logic for its name originally. And then this lower area was called the putamen. And I put the two names here because sometimes I'll fall into the habit of referring to this as the caudate. And in the mouse, we might call it, people often call this the caudate putamen. They join the two names together. So it's hard to escape this in literature. This is part of the Globus Pallidus, which is densely innervated by substance P terminals, so it looks brighter in its immunolabeling. And this is just a rostral part, most anterior part of the Globus Pallidus, which is a relatively large nucleus that you'll see more of. And then one other thing worth seeing down here that I'll talk about more later is the nucleus accumbens we've divided into a core area and a shell area. The shell forms like a little outer capsule around this little angular part of the striatum. It's a part of the habit in the literature is to refer to most of this area as dorsal striatum, and this down below is ventral striatum. And one reason for that is this part was added later as we were able to visualize the chemistry and the connections before we realized that this actually belonged with the rest of this part of the brain. Here we're back. I think we've seen this before. It's redundant. Oops. One other place worth noting is the hypothalamus. This literally means the area beneath the thalamus, like hypodermic is beneath the skin. And here's just a coronal picture where you see where the amygdala lies. And here's the thalamus with the hypothalamus, or part of it below it. But right at the bottom is something called a substantia nigra, literally black substance. And here you can see the globus pallidus is a little larger as we move back. And the hippocampus, we're not going to talk about this much, is right behind the amygdala. Now, this is a picture where we stained, there's a way of in histochemistry staining for iron in the brain. And the globus pallidus is very rich in iron. And you can see the stripes, the fibers that are going back and forth from the cortex and the striatum into the globus pallidus show some of these iron-rich fibers. And you can see the striped appearance of the striatum. The reason we stain this is because in a human brain, this ventral extension of the globus pallidus beneath this fiber bundle here, which is called the anterior commissure, is really hard to visualize. And looking for the ventral pallidum was much harder in the human than it is in animals like rats and mice. And here's the septum, of course, which is a little larger in this particular photo. And here's another view of the human brain stained for iron. You see the globus pallidus and the ventral extension, like a little cup behind the nucleus accumbens. And that relates to its role. The striatum sends projections to the globus pallidus. The ventral striatum, made up of the nucleus accumbens, projects into this ventral pallidum. The globus pallidus extends backward and has axons running through the internal capsule and then runs into the substantia nigra. The globus pallidus and substantia nigra, over the years, people realize that these are really part of one continuous structure that is just interrupted by the fibers of the internal capsule. And then here's this very large picture of the thalamus sitting behind the striatum and to the medial surface of it. And here's a picture of the rat, where things may be a little simpler, but here we have the thalamus, the substantia nigra, the globus pallidus, and then ventral pallidus. And if you see it probably better than I can on my screen, it extends all the way down into this little flat layer here, into this area called the olfactory tubercle. And the reason it sits there is because the olfactory tubercle and the nucleus accumbens are closely related neurochemically and in the pattern of their connections. So just like the striatum projects the globus pallidus, the nucleus accumbens projects the ventral pallidus, the olfactory tubercle projects to this extension that sits in the deeper layers of the nucleus accumbens. So all of the things beneath the anterior commissure here are usually referred to as the ventral pallidum. And the part up here above the anterior commissure is usually referred to as the striatum, but also the dorsal striatum, ventral striatum. Dorsal striatum, at least in the parts relating to motor cortex, are more closely related to the actions and motor actions of the body, and we'll get to that in a little bit. Whereas this part's down here, the ventral striatum receives things from the lower prefrontal cortex, from the olfactory bulb, which is off here someplace, and the amygdala. And it's like, initially it was called like the striatum for the limbic system, processing information related to emotions and learning, rather than just strictly motor learning or procedural learning. So this is another dissection of the human brain, just to give you a picture of the substantia nigra. In a rough dissection of the human brain, the substantia nigra appears black because of pigment that normally occurs in the cells of the dopamine cells that fall in this area. And this black layer is rich in dopamine, and the layer that's a little lighter just beneath it is broken up by dendrites from the larger layer and is more loosely arranged. Oh, and I've misnamed it. This should say pars reticulata. Reticulata mean net-like. So pars compacta, pars reticulata. I'm sorry for the mislabeling. So the pars reticulata looks like Globus Pallidus in neurochemistry and connections. The Substantia nigra pars compacta has this really important group of dopamine cells that are part of a feedback system that goes to striatum. And I've turned this around. You can see this is the dorsal part of the brainstem and the ventral part of the brainstem, whereas in a horizontal picture, these would appear anterior and posterior. Once again, I think we've seen this before. One more picture of the Globus Pallidus, which you see in this particular, I believe, initial stain, in this relatively large structure, the bed nucleus of the stria terminalis. The bed nucleus of stria terminalis gets its name from a fiber bundle that starts here and runs all the way around behind the striatum into the amygdala. Also worth seeing here is the anterior commissure that curves back, goes back toward the amygdala on one side and arcs up toward the frontal cortex. It forms like a little bifurcating wing. And this is a picture of the amygdala. If we talk about the amygdala and you read things in the newspapers, you say, well, the amygdala had something to do with emotion, or the amygdala had something to do with depression, or the amygdala had something to do with heart rate. But the amygdala has a lot of subdivisions. And in fact, it's not doing any favors by referring to it as one large group. We can make some grouping to sort of make it easier for you to understand how this functions. But there's a central nucleus, a central group, there's a medial amygdala, and this large basolateral complex. It's got enough subdivisions to handle all the different functions that people want the amygdala to do. In a human brain, the amygdala, this whole area, is about the size of an almond. It's a flattened oval, so about 1 centimeter across. So that's where it's got its name. Amygdala in Latin is Latin for the Greek word for almond. And we talked about the bed nucleus of stria terminalis. Well, I've tinted this area of the stria terminalis that runs along the medial part of the striatum, around the top lateral edge of the thalamus, and back down into the amygdala. And here's another picture of a monkey brain. And we're going to talk a little bit about something that I call the extension of the amygdala, or the extended amygdala. In our work, we came to realize that the amygdala and the bed nucleus and the stria terminalis are very closely related. They have the same neurotransmitters. They have a lot of the same pattern of connections. But not only that, there's a corridor of neurons that run from one to the other that really make it a solid structure that's just been attenuated by the fibers being drawn up by the growth of the Globus Pallidus and these other structures in development. So here's a Tamarind monkey. This is a small monkey, also in a species that's similar to a marmoset monkey. It's about the size of a cat, but it has a wonderful brain because it's a primate brain, but it's relatively small and easy to handle. This brain is immunostained for a peptide called secretineurin, which is rich in these parts of the brain, but not in the cortex or the striatum, which help us to figure out where things are a little bit, because we can label them, provide a marker for these structures. You can also see it's rich in the hypothalamus, which sits here at the base of the brain, beneath the thalamus. And just so you can see, we're not going to spend a lot of time, but some time talking about the central nucleus of the amygdala and the medial nucleus of the amygdala, or separate structures, slightly different projections. And just like these two subdivisions, the bed nucleus has a lateral part related to the central nucleus and a medial part related to the medial nucleus. We'll talk about their functions in an upcoming section. This is just another view from the cock brain, where you can see this whole corridor from the bed nucleus of the stria terminalis, underneath the globus pallidus, and into the central nucleus of the amygdala. In this case, it's a section a friend of ours, Gary Van Heusen, injected tritiated amino acids, which then follow the axons from one area to the level to the next, and has terminations all throughout this area, again identifying this as a solid structure rather than just axons passing by. So this is a section of a rat brain, and if you're careful when you do the histology and dissect the brain, I've just cut this brain parallel to the stria terminalis. Here you can see the bed nucleus of stria terminalis. In a rat, this is cut at a 45-degree angle to the parasagittal line right here. So, and this is almost exactly the line that the stria terminalis forms in. So you can see this entire structure going around from the bed nucleus to the amygdala, the medial and the central. The thalamus, it wraps around the lateral edge of the thalamus. And then you can see this little gray area are actual cells that follow along the entire length of this corridor. So we're not going to spend a lot of time, but it's good to remember that the amygdala includes the amygdala, extended amygdala, areas that are related to emotion, to the processing of the behaviors we're going to talk about later, include all the cells here, all the cells falling in between, all the cells in the temporal lobe, and then the cells that are up here falling along and following this axon pathway. And historically, it's important because in the 1920s, one of the most anatomists who first pointed this out thought that the bed nucleus of the stria terminalis and this were one structure. If you look at lower animals like fish and amphibia, he thought this lies in one straight line and thought that all of this was pushed up out of the way by the growth of the internal capsule and the striatum. But then he missed this part going beneath, which we sort of revived when we got going in our work. This is just another view. This is the brain of a ground squirrel or a chipmunk, just so you get an idea that the basic outline of the basic plan sits for all the different mammalian animals we can look at. You can see a large optic tract. Chipmunks have color vision. The amygdala, the olfactory tubercle. This white band, if you're used to looking at the brain, this is the olfactory tract, and the band of fibers coming in are the lateral olfactory tract coming beneath the olfactory tubercle and running down along the amygdala. I think this is of high current interest because people, for example, who have COVID virus lose their sense of smell, presumably because the information coming in along this tract is affected, that then can affect this part of the brain, the olfactory ventral striatum and the amygdala. Here we've got the striatum. You can see the striped period of these axons. This is just an unstained brain that's been wet mounted, and then taken a picture with the light shining through it like a shadow. And Globus Pallidus has lots of axons going through. And I like this because you can see the stria terminalis up in the corner with this dense bundle, and then the little gray areas on the other side of this little bundle in the center, which is related to a particular structure down on the amygdala that crosses over around at the bottom, following this arc from one side of the amygdala to the other. So this is a summary of the things we've gone over, color-coded so you don't get confused, of a human brain. So the blue is the caudate putamen, pink is the globus pallidus, the external segment and internal segment that we're not going to spend much time on, and its continuation down into the substantia nigra. Then the yellow and the green represent this extension of the amygdala into the stria terminalis that runs over the top, and the two subdivisions related to the central nucleus and the medial nucleus. And then for viewing pleasure, we have the lateral olfactory tract that goes around in a human on the medial edge of the amygdala and down underneath the ventral striatum and the ventral pallidum. This is a diagram where we've dissected out graphically what the structure might look like in a human, with the lateral part of the bed nucleus, the medial part of the bed nucleus, medial related to medial amygdala, central nucleus to lateral bed nucleus, and this continuous column of cells running across the top. In the rat, this is a diagram that allows me to remind you, we've got the striatum, we've got the globus pallidus, central division and medial division, that the output of the medial division and lateral division differ. Although these are related to the amygdala, The central nucleus tends to favor the lateral hypothalamus. It goes to the brainstem and medulla, while the medial division goes to medial hypothalamus and areas related to the neuroendocrine system. Last of all, I think we've talked about the thalamus and hypothalamus. Just give you a picture of where they're found in the brain, in a human brain. And this is actually, I forgot to mention, this is a wonderful human brain because this is my brain. So I'm giving you a piece of my mind, actually. Again, this is just another way to see the thalamus in a horizontal section with a septum, which at this level becomes quite narrow, and then a horizontal view where you can see the top part of the bed nucleus, the stria terminalis, as it becomes thinner and thinner. Okay, now we're ready for stage 2. So now that you have all these terms, before we go to the next, are there any questions about the terms and where we're going? This is a good time to ask if you've been lost. Nothing? So now we're going to get a little bit more in the nitty-gritty of how the brain's connected. And I'm going to start off at the cortex. And of course, we've talked about the division between sensory cortex and motor cortex. And I've got these pictures just to sort of point out that in cortex, there's a large flow that goes horizontally from initial primary sensory areas like primary visual cortex called V1. And then in visual stream, there's a dorsal stream going on the top and a ventral stream going down into the temporal lobe. And the same thing occurs for the auditory system. Right in this corner here, there's the primary auditory cortex. There's a dorsal stream going over the top and a ventral stream coming down the bottom. And the importance in this is that looking at the physiology and recording the neurons along these two different paths, this path tends to serve recognition and this path place and the integration of visual system with motor features. This one comes down to serve the integration of vision with more complex stimuli, for example, faces, whereas the auditory system up here perhaps goes to things like head orientation and reflexes related to orientation of the body. And this is a gross generalization that you'll hear in no other anatomy class. So the cortex, while oriented horizontally in its flow of information, if you take a cross section, you can see this is just a stain for cell bodies in the cortex and throughout the brain, is oriented in columns that are perpendicular to the surface. And this is the surface in the temporal lobe. And you see this is very easy to see these micro columns and little breaks between them. So one of the great realizations about how the brain was organized came from physiologists who in monkeys and animals who they could record these columns, said if they record around a column, they're grouped together in columns that respond to the same sort of stimuli. So one of the factums of the cortical processing is that little cortical units act like processing units. And these are bundled together in little vertical columns that go across all the layers of the cortex. And in fact, this picture just illustrates their dendrites are even interwoven together, twisted together to make a bundle. And then these bundles and other ones in the near vicinity are thought to work together as a unit, for example, relating things to particular areas of the, in a rat, a favorite is looking particular areas of the whiskers. There's barrel cortex where there's whiskers, individual whiskers in the rats can be found in the cortex and related to barrels of neurons that are vertically oriented in the cortex. The main output of the cortex is these large pyramid-shaped or pyramidal neurons that extend from layer 5 up, dendrites all the way up. I'm not sure it goes all the way to the top, but they're covered with these spines and they get excitatory input from these upper layers. Information from sensory things like the eye, somatosensory system, they come in through these upper layers, layer 4, and the information goes out through this lower layer 5 and 6. And connection between layers come from here and go to like layer 5 up to an upper layer, and things going out of the brain go from layer 5 and 6 down to things like the striatum, back to the thalamus, and then down to the brainstem. So there's a nice organization of the cortex, which is good for a whole nother lecture. So once we get past the cortex, which is represented here, there's a separate organization in the striatum, which is vertical, but we don't have vertical columns. So in the striatum, there's a gazillion amount of these neurons that we call medium spiny neurons. That's because they have medium-sized cell bodies, 15 to 20 microns in diameter, and then this extensive layer of dendrites just covered with what we call dendritic spines. And the spines have the significance that excitatory inputs that when they synapse cause the cell to increase its likelihood of firing or its rate of firing. And the spines seem to be the primary target for that. In this case, the primary target in the striatum from inputs coming from the pyramidal neurons of the cortex or from the thalamus, which is down here someplace, back to the striatum. So when we get back to the lower layer, you remember the globus pallidus, there's a different type of neurons that almost have been, well, they've been called leptodendritic, lepto meaning slender. They have long, slender axons, and they receive a lot of inhibitory input that causes the cell to slow down its firing, because these cells, these medium spiny neurons, use an inhibitory transmitter in a projection from the striatum to the globus pallidus. And it's so interesting that these dendrites form almost like a disc, not for each individual neurons, but in groups that cross a lot of axons coming out of various parts of the striatum and catch their input to focus it on the output of these individual globus pallidus neurons that then go down to the diencephalon in thalamus and brainstem, and then are really the output system toward a loop back to the cortex and a loop that goes out that actually causes some action, things to happen in terms of motor functions. And within the striatum, there's a large group of cells. I'm not going to spend a lot of time talking about them, but theoretically, they're very important. They're similar to the globus pallidus neurons in having long, slender dendrites, but they're not completely free of spines, and they're not oriented like discs in the pathways that cross them. These dendrites collect information and send it locally within the striatum, or in some places, especially in ventral striatum, they project back to cortex. So these feedback loops from these interneurons here back to cortex and striatum, and the feedback loops from thalamus to striatum, and eventually back to cortex, are really key to why this area can handle so many complex parts of information, because every time it goes through the loop, it's reprocessing the same information. In computer programming, this would be like a do loop with an output that keeps adding information, making more and more complex possibilities, which is one reason why this part of the brain can handle complex procedures, which is, at least for the dorsal striatum, one of the roles that's assigned to it. Now, within the striatum, physiologists in the recording said, well, we can't see columns like we can for the cortex, but we can record areas that seem to follow and represent what's coming from the cortex overlying it. And especially when you get down to the pallidus, the globus pallidus areas, there's actually different segments that you can relate to the overlying cortex. There's a lot of condensation of information. If you remember how much cortex sits on top of striatum, as you go from sensory cortex, it has only a small input to striatum, to sensory motor cortex, the places where we get feeling back from the senses of our skin and our position of our muscle, to motor cortex, to the areas of premotor cortex where there's motor planning. The striatum gets larger and larger, and there's more and more integration of input to help to organize these complex motor behaviors. I like to think of it is that when you get well, they don't show visual cortex here. If you get a simple input that's not been conditioned, you get simple responses. For example, a light might cause you to orient toward the light, but you don't have a lot of behaviors to do unless you recognize something in your field of vision that prompts a further action. So as we go from simple stimuli to more complex stimuli, visually, auditory, or even something on your body or sensation, you build up more complex relationships and motor outputs to account for it. So this is from a nice paper in 1986 by Garrett Alexander and his colleagues. And in recording, they say, look, if you look at these columns, there seems to be a column that's focused on the motor cortex, one for the ocular motor cortex that goes to the frontal eye fields, one for the dorsolateral prefrontal parts of the cortex related to motor planning, one related to lateral orbital frontal cortex, which is related to God knows what, perhaps executive planning, they like to say. And then they have this anterior cingulate and on the midline over the septum. And then this projects to what they call ventral striatum, because the concept of the ventral striatum had already been established by then. And ventral striatum gets this anterior cingulate input, which is known to be related or thought to be related to emotions. And then you get input also from the hippocampus, the entorhinal cortex, the superior temporal gyrus, and the inferior temporal gyrus. And what they don't show, you get input from the amygdala to ventral striatum, that goes to ventral pallidum, that goes to medial thalamus. So in fact, this forms a column a lot messier than these nice ones that are related to part of your hand or part of your arm. Some of the physiologists said you can come to the hand area of cortex and you record a hand area from the proper part of globus pallidus, but you can't get to the area anterior cingulate and point out an area. You can follow this projection, but the emotions and the things that are being handled are complex. It's hard to pin this down with an electrode and come up with a particular individual stimulus. So about the same time, this is really from papers we're writing in 87, 88. We said, well, look, if you look at, this is the column they talked about with the anterior cingulate cortex, where you go into ventral striatum, which we knew, and me and my colleagues, Leonard Hamer and Jose de Olmos, Leonard Hamer had originated the idea of ventral striatum. So this is the column of ventral striatum that was the last column we talked about in the preceding picture. And this is the striatal pallidum system, which had all these other columns that we saw in the preceding picture. Well, this is like striatal columns, but you could include the amygdala, which the Alexander and all did not. And if you look the septum and the diagonal band on the midline, excuse me, do the same thing for hippocampal formation. They go to the lateral septum, and you go to the diagonal lateral septum and diagonal band. So I saw this, and when I saw this and wrote about it, I thought this was wonderful, because it's made like a game plan for understanding the organization of the entire forebrain. And I was so happy you could hardly believe it. So if you keep that organization in mind, at least in this section, I'm going to give you a little bit further picture of what this looks like in the rat. And coming back to the pictures we looked at in the first section, just so when you see it, you'll recognize it, because it's related to what people have been able to study. And as they've gradually accepted this idea, when they're studying different parts of the brain, they started to relate, oh yeah, when I study the bed nucleus, I should also study the central nucleus, the lateral part of the bed nucleus. When I'm studying the medial part of the bed nucleus, I might see something like the medial parts of the amygdala. The blue cells here marked in dots simply relate part of the interneuronal system that you can see for the extended amygdala, the extension of the amygdala into the forebrain, that are similar to the interneuron system that you can see for the dorsal striatum and ventral striatum, except that these neurons project all the way around the frontal parts of cortex, and were thought to be really important for tying together learning in relationship to the output of these various outputs of the amygdala and ventral striatum. The ventral striatum projects to parts of these large cholinergic, CAT stands for cholinacetyltransferase containing neurons, These cholinergic neurons are easy to identify by virtue of this enzyme that they contain. And so part of this topography, you get ventral striatum coming in here, you got amygdala coming in here, providing feedback related to processing of autonomic system, emotions, and things related to visceral sensory information, feeding back to their appropriate parts of the cortex. And just so you can get an idea of what it looks like in the rat, I've immunostained this brain in a rat for angiotensin 2, which is a peptide, it's a hormone, and it's a transmitter. There's a horizontal section in the rat, there's anterior, posterior, and angiotensin 2, if you think about our current situation, the COVID virus attaches itself to a protein that's called the angiotensin 2 converting enzyme 2 in the body and the brain. The angiotensin 2 converting enzyme is part of a protein complex that's on a receptor for angiotensin 2 called the AT1, the angiotensin 1 receptor. And so when these spikes, they have a shape on the end of the spikes, they attach to the receptor. And normally when angiotensin attaches to the receptors, the cells take the angiotensin and that part of the membrane rotates inside where angiotensin is broken down to smaller peptides and cycled to angiotensin. There's actually a name for this peptide, angiotensin 1,7, which by itself can be released and have biological actions. Angiotensin 2 circulates throughout the body. It gets its name from the fact that when they first discovered this peptide, which is released, prompted by a release of chemicals from the kidney, it increases blood pressure. Angio for blood, tensin for pressure. So angiotensin is a potent molecule causing an increase in blood pressure. But in the brain, what it's doing in the hypothalamus and the amygdala is likely to be something related to other functions and not strictly blood pressure, although these areas do have connections back to the nucleus of the solitary tract, which also has angiotensin, which you can see in its horizontal section, all the way back in the brainstem in the medulla. So at least part of the message is that when you talk about what's going on with angiotensin, you have to worry if it gets into the brain where they would have a preference for these parts of the brain that have angiotensin normally. Hopefully the brain has a blood-brain barrier that keeps it out, but some of these peptides and some of these viruses have a way of latching onto axons and be transported into the brain, what's called neurotrophic virus. Polio virus is neurotrophic. There's one other, some of the herpes simplex virus is neurotrophic, that get into the brain, they're slow to cause damage, but the damage they cause can be pretty devastating. This is just a schematic so you can see how these things are interwoven in a rat brain where we can see things. There's dorsal pallidus and ventral pallidum, and then extended amygdala works its way, squeezes around this particular structure. And here's a similar immunostaining. In this case, the red is stained with a compound called epidermal growth factor, which is stained densely in Globus Pallidus areas, but not in the striatum and not in the amygdala. And here we have the angiotensin 2 staining. It stains in the amygdala, but not in the Globus Pallidus or in the striatum. And here, this is the internal pallid segment equivalent for the rat. In older days, they used to call this the entopeduncular nucleus, or the nucleus within the cerebral peduncle or internal capsule. Now the atlas, people who draw maps of the rat brain have gotten to calling this the medial pallidal segment of the rat. So here's a diagram so you can follow up what's going on in the amygdala and its connections. Now, this is a horizontal view. If you go back here, a horizontal view in a diagram where this is the anterior and posterior, and I've laid it out horizontally, except I've rotated it. So this is anterior, this is posterior, this is lateral, and this is medial. Just like we could see those columns labeled of extended amygdala, this is this extension of the amygdala, but that's not all of the amygdala. There's a large lateral cortical complex that doesn't stain for angiotensin 2, which we've argued is more like cortex than it is like the centromedial division. And like more like cortex, I mean it has pyramidal neurons and processes information to the medium spine or the spiny pyramidal neurons that are the output neurons is an excitatory transmitter, and they project inward to these other divisions of the amygdala, but also to the striatum. This part here is called, well, the cortical superficial nuclei of the amygdala. I've taken this and put like a little hinge here and flipped it over and rotated it so I get all the nuclei in one plane. But this would be its actual position if you're looking at the brain from the bottom, sitting on top of these other deeper lateral complex and part of the medial complex and central complex of the amygdala. So when you look at the amygdala, we're talking about the way these things process information. All of the overlying cortex, in a sense, the lateral complex of the amygdala, the lateral nucleus, is at the end of this visual horizontal stream of information. It's not the only place at the end of this horizontal stream of information, but it relays information from the cortex to the lateral nucleus and into this basolateral complex. There's an anterior and posterior part, a medial part, and these other areas, which are the other subdivisions of the amygdala. I don't know, this is something called the amygdala piriform transition area, amygdala hippocampal area, and we're not going to talk any more about them other than to know that they're part of cortical-like nuclei. And here you can see these medial divisions isolated. There's one structure here we haven't talked much about, which has this wonderful name, the interstitial nucleus of the posterior limb of the anterior commissure. If you remember, the anterior commissure has axons running back into the amygdala. Well, it separates the medial part of the central division from this far lateral division, and the globus pallidus pokes its way through here. And because of the histochemistry and the connections, we're fairly sure that this belongs to the rest of the amygdala. But we've kept a name because my friend Jose, who studied it initially, named it back in the beginnings of the 1970s because he thought this was related to neurons following along this bundle of axons, but he thought it belonged to the striatum. And since then, in the late 70s, it's actually my work mostly and my colleagues in Virginia. We've shown that the neurons and the axon and neurochemistry forces this into the amygdala. Here's a separate division of the medial amygdala, where all of the anterior basolateral amygdala projects to striatum. Posterior part of the basolateral projects to this extended of amygdala. Uh-oh, are we out of time?
No, you're good.
Oh. I've just lost my picture of what I'm looking at. Susan?
I can see it, George.
I think we're seeing it.
You're seeing it, but I can't see it.
Okay.
A little tougher. I can.
No. Let me just see if something was hit on your computer. You're pretty familiar with the setup.
Well, the projection screen went up.
Oh.
It may still be there, but I can't see it. I need a little drink anyway. I warned Kim that I get a little hoarse because I have allergies. It has nothing to do with COVID. It has to do with how much my voice gets used in the middle of the watch your head.
This is really amazing.
Did we blow a fuse? Where's the picture? Oh, good. I might be able to get back on my screen if I had to. Nothing? I can do it. I think it's loose. Here it comes. Maybe I cut the wire. Okay. So now you've learned what we've talked about, and what you learned in this first hour is practically everything it would take a whole semester to teach people about. But I've made some pretty awful generalizations in doing this. This is just a picture to help you visualize. Remember we talked about the lateral complex to the amygdala and the central and medial complex. Part of why we think this belongs with cortex is that it has the same chemistry as cortex and has the same cell types. And this is just an illustration of it using an immunolabeling for an enzyme that's called glutamic acid decarboxylase, which is an enzyme that's in a step converting glutamate into GABA. And GABA is, what is GABA? Gamma aminobutyric acid is one of the main inhibitory transmitters in the brain. So we can use the immunohistochemistry to get a hint on how some of these subdivisions fall out. So here's just a little reiteration so you can see the view of how these subdivisions, especially EPAC, goes around the outside of the ventral pallidum, and there's a medial corridor of the central division and medial division that go below the globus pallidus. So we're so fortunate. I don't know if you've been following, but in modern times, the biochemistry that the geneticists have worked out have allowed us to see wonderful things. In this particular mouse brain, the neurochemist has worked out, taken an egg and injected a construct of a protein that is expressed carrying a fluorescent protein, but only in cells that have this enzyme, glutamic acid decarboxylase. And when these mice are born, if you shine an ultraviolet light on them, you see these neurons all over the place, especially in places, because their brain when they're born is fairly transparent, and so is their skull. And in this particular mouse species, by changing the genetics of how you affect the embryo of these cells, the egg, and how you insert this into their genetic code, you can attach this green fluorescent protein and now a red fluorescent protein with any number of other proteins that interest you in the brain and, for that matter, elsewhere in the body in multiple colors, because now there's a green fluorescent protein, there's a red fluorescent protein, and there's a blue fluorescent protein. And so we can color the brain when the organism is born. And in this case, this mouse is growing up, and then we sacrifice the mouse, cut the brain, and we look, there are the fluorescent neurons. We just have to have the right type of microscope with a fluorescent excitation and a filter to view this. Now, I was already in Chicago, and a friend of mine, Gabriela Serkova, who was a Hungarian from Slovakia, was a postdoctoral fellow in my friend's lab. And she was looking at this, showing me this brain. I said, holy smokes, this part of the brain, accidentally, she was going to throw this brain away because these neurons that contain this glutamic acid decarboxylase are actually found all the way in the striatum and in the cortex. And this was, and to some degree, in the thalamus. And she was going to dump this mouse strain because it didn't label these parts of the brain where it should. But in labeling structures in the brain, some of the flanking peptides, nucleotides that determine where a gene is expressed in the body can determine its localization. And when you make these things, you're never sure. You sample ones to cover all the different areas you're interested in looking at to have this enzyme. But this one had flanking peptides that focused it on parts of the hypothalamus, but also the medial division of amygdala. And I love it because it gives you just a good view of what this looks like. In the posterior part, there's several columns. There's a dorsal column that comes down here, an intermediate column with lots of GABA cells, and then a more lateral column, all belonging to the medial part of the bed nucleus of the stria terminalis. The medial part of the bed nucleus of stria terminalis, along with the medial amygdala, are important because they project to medial hypothalamus and help control the pituitary, but also project down further to things in the brain related to reproduction and copulation and all kinds of things that we know and love. And as we predicted, because these neurons label the medial amygdala, they also label this corridor going beneath the globus pallidus, the medial part of the sublenticular extended amygdala. Sublenticular because it's beneath the lens-shaped nuclei formed by the globus pallidus and the overlying dorsal striatum, and doesn't label the central division of the sublenticular extended amygdala. So before she could do anything with these mice, I got busy and took a whole picture of what this looked like, because I figured I might not see this again. And I don't think I've ever published it, but I've shared it with my colleagues whenever I give a talk. If you spend enough time looking around at the brain, you can see this little shadowy area that doesn't have these neurons. I actually don't remember whether it should, but this is a cortical-like nucleus, but it's called the nucleus of the lateral olfactory tract. Those olfactory axons we talked about coming right at the base, and then the neurons here collect, and they send their axons into the stria terminalis. If you remember that little bundle I pointed out in the stria terminalis, this one goes all the way across and across the commissure to the amygdala on the opposite side. And it's called the commissural component of the stria terminalis. It is very easy to see in mammals because it's always in that little bundle at the base of the stria terminalis. And I'm not going to talk about that anymore, but I like this nucleus and couldn't help pointing it out to you. So again, this is just another picture. I think I've shown this before with secreted neuron, with the ventral columns between the medial amygdala and the bed nucleus, and some of the staining in the lateral septum, which is not unrelated. And this is a picture to remind you, to tell you about the human brain. If you want to study these areas in the ventral striatum and this extension of the amygdala, striatum and globus pallidus are monsters. They're wonderful to study. But this part here is all mashed up in a human brain. It's the worst place to study it. If you look at a monkey, this is a piece of cake. If you look in the human, this part, you see this part of cortex in the human brain. If you remember the earlier sections we showed you, the cortex comes and lies underneath the ventral striatum, because the frontal part of cortex, as it expands, it gets bigger and bigger in humans, starts forcing it away until it causes that whole part of the brain to rotate, and everything down in this area gets smashed, and they've got to find their way through the best they can. So it's a disaster. We can find it by using immunostaining for peptides. This is staining for the peptide vasoactive intestinal peptide, which their names are sometimes related to what people thought their hormonal function was. But when you get in the brain, all bets are off. And you can see it's densely labeled in this dorsolateral portion of the bed nucleus of stria terminalis. And little islands that are interrupted, it actually wound up flying back into this little compact part of the central nucleus that's surrounded by a larger part, but shows that these two parts are really comparable in their immunostaining, and their cells and their connections are comparable. If you look at the staining for this peptide, neuropeptide Y, you get just the opposite in mirror. This part, the dorsal part of the lateral bed nucleus of stria terminalis, doesn't stain very well for a neuropeptide at all, but this little central corridor also doesn't. These guys have paired staining, which makes us think that we're looking like a parallel corridor where there are matched components at either end of this dumbbell-like structure. Then, of course, there's little islands of things that are confusing as hell, trying to work their way around back from one structure to the other. And then, of course, this large arc of axons and neurons going over the top, connecting the two. So it's a large structure in a human, but it doesn't have the simple continuity that we can see in rats and smaller primates, including the macaque. So rat, easy to observe, but then we have to extend it to the human, harder to observe, but we know the chemicals that are there, we know the connections, and now the connections of the brain are getting easier to study, so we can make the homologous areas and identify them easier between one species and another. So what comes after this? After this is the reminder. I told you before that if you look at the medial part of the central, and the central nucleus division of the extended amygdala, they have slightly different outputs. Well, here they're horrifyingly projected for the medial division, which has projections to the thalamus, interconnecting connections from the medial amygdala to the medial bed nucleus and then down to medial hypothalamus. And the output, however, of the central division is pretty extensive. It goes out to the lateral hypothalamus, paraventricular nucleus of the thalamus, to the near brainstem and the rostral brainstem, into the medulla, all the way back down to the nucleus of the solitary tract, and to autonomic columns in the ventral part of the medulla. This is one busy part of the brain, which is one thing that distributes the columns for the amygdala between those of the striatum, which are much simpler. If we look at the projections coming out of the striatum, this is an injection of an anterograde tracer. Sometimes a history of this, mechanics of this, would be fun for a talk, because when I started, looking at connections in the brain was really hard. The best we had was like a degeneration. You could make a lesion or a knife cut, destroy part of the brain. And if you're lucky, you could impregnate the axons with a silver-containing stain and try to follow them through the brain. But here we've injected a protein called Phaseolus vulgaris agglutinin, which everybody calls PHAL because the name is really a tongue twister. This is, I think, maybe derived from peanuts, but this protein sticks to membranes. And so if you inject it into the brain, it sticks to the cell bodies that are there, but then it fills up the cell bodies and its axons so you can follow the axons wherever they go in the brain. So we were talking about the projections of striatum. This is the core, part of the core of the nucleus accumbens. And you remember we talked about the fact that the striatum has vertical columns that are topographically organized. Well, for the core of the nucleus accumbens, this topography is sort of the same. Lateral part of the core goes to lateral part of the ventral pallidum and to a lateral part of the entopedunclear or ventrolateral part of the entopedunclear nucleus. If you were to inject the medial part of the ventral, of the core of the ventral striatum, which we have, we project to an area just more medially and to another area more medially to the entopedunclear nucleus. But if you inject the amygdala, or the extended amygdala, in this case EPAC, which is like the central nucleus of the amygdala, you get axons just going all over the place and branching like crazy in the lateral hypothalamus, interconnecting the lateral bed nucleus with the central nucleus, and the interconnecting EPAC, and then going down to the brainstem, making this large network. So this is, we showed the central nucleus before. This was part of the study that we published, showing this lateral column is like the central division with most of the same projections to lateral hypothalamus, to the pontine tegmentum, and then to nucleus of solitary tract, and down by the, actually down below the nucleus ambiguous. The picture I showed you was my favorite. And in contrast, there is a part of this lateral, it's still called EPAC, that in the course of our studies came to be realized was a lot more like the shell of the nucleus accumbens, well, like the shell and the core of the accumbens, had a pretty much typical output related to the striatum rather than an output that was characteristic of the pallidum. So here's a rather focused, narrow corridor of output. Here's a wide diffuse output related to visceromotor, complex frontal areas related to executive planning, emotions, and God knows what else. Reproduction, parenting, and all the rest, which might be part of the next section if we get to it. The last thing, I think we touched on the fact that you can make a division between the core and the shell of the nucleus accumbens. This is just a pictures from a paper we published in '85, where we were staining for this peptide cholecystokinin, it's a wonderful name, which densely stains in the shell, the medial parts of the shell, not the lateral part of the shell of the accumbens, and not in the core of the accumbens. This is just acetylcholinesterase which has a slight difference between the shell and the core, but you can see the continuity of the dorsal striatum and the ventral striatum. And acetylcholinesterase is an enzyme that helps break down the neurotransmitter acetylcholine by carving it in two, hence the name esterase. But it was one of the early stains that allowed us to see the similarity between the dorsal striatum and the ventral striatum. So in this area, we actually coined the term between the core and the shell of the accumbens. If we get to it, probably we will. Here's the fact that you can see the core and the shell of the accumbens in a monkey brain. And this is a reminder that it seems to merge a little bit with the bed nucleus of the stria terminalis, and may well be because the bed nucleus sends axons into the shell areas of the accumbens, pulling it in a little closer relationship to the amygdala than the rest of ventral striatum. There's a horizontal picture in a Tamarind monkey, just showing the staining for the secreted neuron, which stains densely in the amygdala areas. You can ask me about this part later. Too fancy to cover. And so this is just a demonstration in the rat. When you put this tracer, PHAL, that labels axons in the bed nucleus of stria terminalis, you can also dense labeling in the shell of the nucleus accumbens, but not in this core, which is pretty rostral here, where they're pretty well attenuated. And in terms of the connections, that's about where we are. And I think I have time to go into a section on at least how these functions are impacted by these divisions. This is a reminder, this is a photograph whose name I should remember, but it's in an art museum in Boston, to remember that these are my main collaborators. Leonard Hamer was a Swedish neuroanatomist that I started as a postdoctoral fellow in Denmark, where he was a professor. He had been a professor in Virginia. I had made a postdoctoral fellowship application to work with him. In the meantime, he moved to Denmark to take a position there. After I was there for a year, he moved back to Virginia. Jose had been a postdoctoral fellow in Leonard's laboratory since the early 1970s. And he came to work back worked in Denmark, but he also came back to work with us periodically. Every year he came back from Argentina, where he was from, and worked with us in Virginia. Laszlo Zaborski and Scott Zahm were postdoctoral fellows who came into our lab. Laszlo eventually became a professor in Rutgers University. And Laszlo was important for staining the core and the shell of the accumbens with cystokinin. He was an anatomist, studied hypothalamus in Hungary, and eventually immigrated to the United States. He worked with us for 8 or 9 years and went on to Rutgers and has become an expert in this basal forebrain, and especially in these cortical pedal areas of the brain, but he's also a long-term expert in the hypothalamus. Scott continued to work in ventral striatum and is now at the University of St. Louis. And Professor Sarah Shabalanyado was a professor who took a senior postdoctoral fellowship in Virginia, was a senior collaborator, a hardworking woman, about 5'2", but a bundle of energy, who worked with us, was a very strong collaborator for 6 or 7 years, even after she returned to Brazil, and helped develop some of the structures. We were examining the interstitial nucleus as an extension of the Globus Pallidus. She ran through that parts of the brain like a train. You don't have to suggest what was going on. She worked until it hurt. And we have to give these people the credit that deserves. If this is like me looking through the microscope, this is the importance of the collaborator telling you what to look, where to look, and what might be important. So this is dedicated to all those collaborators. I might offer a break right now, but are there any questions before? I know that was a lot, and if you're asleep or you've left, I'm sorry for dumping it all on you, but this covers a lot of the brain that may or may not be useful for you to understand what might be going on. What do you think, Kim? Anybody?
This is Louis Morel. I have two questions that are related to each other. They have to do with staining. You mentioned early on that it is possible to create antibodies to some of these structures. And is that the same as immunolabeling? And more importantly, whether they are or not the same, can you describe the techniques used and what compounds are used and what serves as an antigen for these antibodies?
So this is actually very similar to making a vaccine. When you're trying to make a vaccine, what you have to do is isolate a protein and get a pure compound. So that's the first step. When I started, immunostaining for the brain wasn't very helpful. The first thing they were making antibodies for vaccines. But one of the first things they were doing was making antibodies to small molecules that they wanted to analyze. And antibodies could bind these molecules and help to isolate them. And if they're radioactive, you could sample radioactivity and do quantitative measure. So they had what they call was a radioimmunoassay. So people making vaccines and trying to make antibodies to these different small molecules got to be very good at making antibodies to specific structures. But you had to have the protein. You had to have the protein and the way it was folded when you find it in the brain. If you had that, you could inject rabbits, sometimes rats, and later on mice as the technology developed and get the antibody. This is no different than making an antibody for a vaccination, except in this case, we want to have an antibody that responds to a peptide. There's two things. You want an antibody that responds to a peptide or a protein, but it has to respond to a peptide or protein in the area where you're looking. And oftentimes for us working in the brain, you want for one that is an antibody that'll stain it or bind to it when it's fixed by formaldehydes and things. It's tougher than it sounds. The first people developing this technique used fresh brain tissue, bound the antibodies, and then dipped the slides in fixative afterwards. But later, as people got better and better, they found ways to minimally fix the tissue, float the sections, and attach the antibody to it. Well, attaching to the antibody doesn't do you much good unless there's a way to see it. So what they did is probably around the last part of the 60s and the early 1970s, they developed a sandwich technique. So they developed an antibody to antibodies. This is really nice, so that they know in an antibody, the part of the structure recognizes the protein or the peptide or the molecule you're trying to see. The other part is a long tail that's general for all the antibodies that are made by a rat. And I can remember there's called FA, FB, and FC. It's like a little Y-shaped molecule. Well, if you make a molecule an antibody that recognizes this other antibody, you can play some funny tricks. You can use that in a sandwich to attach some marker to this other last antibody, and then use that. In the beginning, they used fluorescent compounds. Fluorescein, which you sometimes get dipped in your eye at the optometrist, could be chemically bound to that second antibody. So if you get the brain, you can primarily label the first antibody, but that's really expensive. They were the most expensive antibodies. But the secondary antibodies you can make in great batches, they were cheaper, so you label the secondary antibody. So this sandwich technique was developed from this idea of having a marker on a second antibody that was fluorescent. After that, someone came up with yet another idea that you could use the second antibody to link to an enzyme that could catalyze a reaction that can allow the deposition of an insoluble marker that would be precipitated at the spot in the brain. This is almost worth another talk. I'm not going to go on, but you can make sections precipitate a chemical. Most commonly used chemical is diaminobenzidine. And benzidine, when the reaction is finished with the enzyme called horseradish peroxidase, it makes peroxide, and benzidine makes what's it called? It makes a polymer. Benzene forms a polymer, which is insoluble, drops at the brain. It's also light dense and electron dense. So then you can look at the brain. So a lot of the sections that were stained like dark brown versus light brown, that was this dark brown precipitation of the polymer of diaminobenzidine. I hope that answers your question, or
Well, it does help. Thank you.
Yep.
Yes.
George, we've got about 10 or 12 minutes left. So I don't see any more questions in the chat area.
Let me get, if you want, I can give you two things about the behaviors that are important. And I can give you a quick section. There's about 3 or 4 sections I've attached here that relate just to the importance of this part of the brain. And let me go through that just to give you a flavor, because that would be a better stimulus for more questions afterwards, okay? The first one is that we talked about dopamine coming from substantia nigra to the striatum and ventral striatum. At one point, probably about the late 1960s, as the neurochemists could start to measure dopamine in the brain, they realized that dopamine is crucial for addictive drugs. Almost every drug that's rewarding, produces a high, impacts this dopamine pathway. This is just a picture of an animal with electrodes in the dopamine pathways. You give this animal an electric shock, and he'll press a lever, he'll run to a certain part of the cage, or in this case, he'll lick and even eat for the pleasure of getting this electrical stimulation in the brain. The reason this is important is the dopamine receptors in the brain have become the focus of where addictive drugs work. In this particular case, you can image radioactivity in the brain using positron emission tomography, which is a big machine measuring the radioactive output of a radioactive compound you've attached to a molecule that accepts to a dopamine receptor in the brain. And you can watch how this receptor fades between higher doses, and labels, if you should recognize by now, the ventral striatum, nucleus accumbens at high and lower levels depending upon the concentration. This molecule is blocked by preloading this brain with things that bind the receptor before they get there. So for example, if you have a dopamine agonist like amphetamine, you can give the human being amphetamine, and if they have high binding of amphetamine, they'll have a lower binding of this particular compound, raclopride, which is a lower affinity receptor for the receptor. Now, the interesting part is you've compared people on how they respond to high to the dose of in this case, methylphenidate. The higher the binding, the greater the high. This is a direct image by Nora Volko, who's now actually section chief of the National Institute for Drug Abuse, imaging the compounds in the human brain and how they affect imaging in addicted and non-addicted people. So one of the theories on why addiction occurs is that when you're exposed to addictive drugs like amphetamine, the receptors that receive it are downregulated because you're hitting it with a lot of compounds that affect those receptors. And it sort of said, well, I need to balance things out, so it downloads a receptor. The problem is that when you don't get the amphetamine, the rewarding effect of normal stimuli has also been decreased. You can't get that stimulus because you have fewer receptors. One of the reasons there's a withdrawal when you're doing looking at drug addicted patients. And this is just a picture of the binding in a normal patient. And this little dot, which you now recognize, is in the ventral striatum and close to the rostral, to pull to the bed nucleus of stria terminalis, much lower in a brain of a person who's addicted to cocaine, I think it was. And then you could plot the behavioral ratings by giving them methylphenidate in terms of craving versus the binding, because you can block the binding of the amphetamine. The higher the binding of the raclopride, which blocks the amphetamine, the higher the binding of the amphetamine reduces the craving, the lower the binding in the brain. I may have this screwed up, but it's complicated. But we can match the binding of chemicals in the brain of a human to their chemical effect in the brain. And using imaging now, we can see where this is happening in the brain and compare it between normal and drug addictive individuals. You remember I showed you this diagram of the extended amygdala, that this part of the brain projects the striatum, part of the amygdala projects the striatum, and this part projects to the extended amygdala. There's an argument when you look at drugs if there's an unconditional effect of a drug that makes you feel good, you get the rewarding effect. But there's also a conditioned part. If you remember Pavlov and his dog, there's a conditioned stimulus can make you do things if it's hit a lever or something in order to get the delivery of food into their mouth. Or in this case, you can have rats conditioned to hit a lever or go to a certain place in an open space in order to get a stimulation in the dopamine bundle or to get an injection of amphetamine. If you take a blocker, in this case, I think, I don't care. If you put a blocker into the anterior part of this lateral complex of the amygdala, you can block the performance for that rewarding drug effect. Oh, wrong again. If you put the, yeah, here we go. If you block this, you can block the performance for the cues that sustain the behavior. If you block this part of the brain, you block the responding for the drug. So the fact that there's responding for the conditioned stimulus, the light that goes on and says you can press a lever and get the drug, is related to this information coming from the anterior part of the basolateral and going to the learned behaviors, because that's one thing the striatum and the ventral striatum seem to be doing, is learning procedures and behaviors that help us organize our daily life, like drive a car to get someplace. It's purposeful behavior, but it's somewhat automatic. And then the actual stimulus of the drug itself goes to this part of the brain, which feeds back and locks in, projects back down to the dopamine cells, fires up the motor system, and says, hey, that was important. You should learn about this and what's going on. So the anatomy and the behavior and the drug taking seem to fit together in a really swell fashion. That's the part for this section. What do we have here? There's two other possibilities. One, oh, I know what we want. Given the time, we were just talking about drug. You take a drug like methamphetamine and inject it in a rat. Anatomist had a procedure for staining degenerating terminals selectively. If you make a single dose of amphetamine, it causes degeneration in the striatum, which is a section of the rat brain here. Here's a Globus Pallidus striatum, cortex. So it's awful. We get one dose of amphetamine, you get degeneration in the brain. Now, it's been argued that one reason for this is it changes their body temperature and all that. But the funny thing is it doesn't cause degeneration in the related parts of the amygdala. There's dorsal striatum. Here's an area around EPAC, the interstitial nucleus, where the terminals are very few, hardly stained. Here's a normal brain. It's a little dirty, but you don't have this terminal degeneration in this comparable area or an EPAC. If you go back to the core and the shell of the accumbens, the core of the accumbens looks like this. The shell looks like this. So the part of the brain that sustains responding for drug abuse is highly focused on the ventral striatum, but even more so on the shell of the accumbens and the related parts of the amygdala. They don't get degenerated, generating terminals by exposure to compounds that overstimulate the system. The striatum and the dorsal striatum and the core of the accumbens does. And in a distant way, you might relate to damage to the dorsal striatum can cause compulsive behaviors in a different kind of pathology. So this is at least one type of pathology that's a little worrisome for drugs of abuse. Here's the second part. This is the brain of a schizophrenic. It is very dense staining, is gliosis. This is not the normal picture for a schizophrenic, but it's a reminder if you get damage to this part of the brain that's this bad, before they ever looked at this person's individual brain, when he was alive, he behaved like a schizophrenic. So if you damage the amygdala, the parts in between, maybe not the bed nucleus, by whatever reason, through a stroke, between transport of a virus or something into the brain, you're going to seriously disturb behavior. And that includes in Parkinson's disease. So there's a very that's not completely accepted, let's say, well thought of theory in neuropathology, that if you look at staining for Lewy bodies, which is a type of pathology that was identified early on in the brain of Parkinson's disease. This is the brain of a person who was diagnosed with Parkinson's disease, and all of the brown staining here are Lewy bodies, which are dense in the cortex and the striatum and substantia nigra. But if you just look at the staining for Lewy bodies, which in modern times we can identify because there's a certain peptide, secretoglucan or protein, I can't remember which it is, that is disrupted in familiar genetically based family diseases of Parkinson's. This secretoglucan Pol molecule is always found within Lewy bodies. So as a proxy for the histochemical tricks or silver stains used before, you can use the immunolabeling for secretoglucan. And if you start saving normal brain, you can see there's pathology that turns up very early on in the centromedial amygdala. See up here in part the bed nucleus. So in very early stage 3 and stage 4, hardly anything, but these parts of the brain occur early. The pathologist who turned out and pointed this out also stained the brainstem. The first places in the brain that get stained are in the medulla that get afferents from the periphery, from the peripheral nervous system, and send afferents back to the peripheral nervous system, to the gut, to the heart, to the lungs. And his theory was, if you go down and look in the peripheral nervous system of deceased individuals, you start to see these Lewy bodies in the peripheral nervous system even before you can find them in the brain. So his idea is that these toxins are retrogradely transported into the medulla, from the medulla to the forebrain, for the forebrain, and the medulla basically in a cascade that goes throughout the brain. And this type of transport is not unheard for neurotrophic viruses. It actually has been shown if you take the secretoglucan compound in experimental animals and inject it in one part of the brain, it causes degeneration, but then gets picked up and transported to another part of the brain without being disrupted or destroyed. So in fact, this kind of transported neural death is very high on the mind these days of neuropathologists for diseases like Parkinson's disease, and also in, here's COVID-19. And I've cherry-picked the samples to make it easy to see what's going on. Now that you've seen these structures in the brain, these are individuals. This is that one woman who's died of COVID-19. I think she's dead, actually. I'm not sure now. But you can see in the centromedial amygdala is hypodense because when the tissue starts to degenerate, it has more water, produces a lighter signal in scanning MRI. And then the dorsal thalamus was heavy labeled, but you can also see now that you've been educated, this little wisp of the bed nucleus of the stria terminalis extending into the back end of the nucleus accumbens. And I put the arrow there because the pathologist didn't even point this out because he didn't know what it was. Here are two other individuals who are stained. You see dense, hypodense areas in the frontal cortex, in the amygdala hippocampal area, and in another brain, there's degeneration off here in the layers of cortex, not much in thalamus, but this little area of bed nucleus of stria terminalis accumbens. And the idea is these areas are stained early on because of their dense connections to the brainstem, in the brainstem nuclei that are innervated by these amygdala, especially the central division. You have the facial nucleus, the motor nucleus of the trigeminal nerve that goes to the nose and the face. Hypoglossal nucleus projects to the tongue. You got neurons in the nucleus ambiguus that project to the larynx and the throat. Neurons in the Tract solitarius that project to the lungs and to the blood vessels, some of the blood vessels, to cause modulation of homeostasis in the body, regulation of breathing, regulation of heart rate. When these areas are affected by a virus that affects the neurons, they can get transported back to the cell bodies. If they reproduce there, they can stay alive inside the cell, or they can destroy the cell, picked up like a cascade into the brain. If they destroy the neurons very quickly, you're in big trouble. In some viruses, like rabies virus, transports very slowly, doesn't kill neurons, but gradually can go from the stomach to the brainstem to the the forebrain to the amygdala to the cortex, but slowly it'll cause degeneration. So if most people maybe don't appreciate this, if you get bit by a rabid dog or a rabid raccoon, it could be a year later before you show the symptoms. If you don't get a vaccination to snuff out these viruses, I think it's a virus. Yeah, these viruses that are being transported slowly throughout the brain. The rabies virus is so useful in tract tracing. People have taken rabies virus of a particular character that you inject in the stomach, so you can see the parts of the brain in a rat or a human that projects to the stomach because it goes across the different synapses in the brain. And this is an example of
I just wanted to, because I know that you don't have a clock in there. We are at 2:30, and I just want to make sure that before you either move on, if you have a few more minutes and people are staying, I want you to continue, but I just wanted to make sure that you are aware of the time.
Okay, I was having such a good time, I was losing it.
We're
But
having a good
I think
time.
this is the last section in the series.
Okay.
This should be brighter for you than it is for me, but this is a section of my own from a rat where I injected the stomach, or actually my friend injected the stomach, but I've got the brain. Injected the stomach with a pseudorabies virus. This is an alpha herpes virus that causes disease in pigs, sometimes called swine virus, but doesn't really intoxicate humans. And you can see the central nucleus here in the right timing is really nicely labeled. There's a medial part of the central and a lateral part of the central. You remember we talked about how this is like striatum. The lateral part of the striatum has medium spiny neurons. The medium part of the central has pallidal-like neurons, these long dendrites. And this is the paraventricular nucleus of the hypothalamus and its extension over the lateral hypothalamus, which also projects down to the region of the nucleus of solitary tract. It has motor neurons that project to the wall of the stomach, and for that matter, down to the upper gut in the stomach. And then more neurons down here related to the hypothalamus. So by taking advantage of these disease vectors, what we call neurotrophic virus, we can study the brain, but it also means when the brains come along, we can worry about where they're coming. In other pictures, I've shown you the angiotensin II labeling in combination with the Globus Pallidus. Just a reminder, when you look at COVID virus, it's staining receptors, the AT1 receptor for the angiotensin II. Well, this is where angiotensin II is in the brain. The AT1 receptor is not everywhere that angiotensin 1 is. Otherwise, they wouldn't just call it angiotensin 1. There could be an angiotensin 1 receptor and angiotensin 2 receptor. So depending upon where this receptor is the place where you'd expect to find the binding of COVID virus that binds to the angiotensin, binds to the angiotensin converting enzyme, which is part of the AT1 receptor. So this may give you a hint of why when we looked at those COVID brains, up in these areas we get it, degeneration, where we don't get it in the related areas that are close by in striatum. And that's my other favorite neuron. I'll tell you about that. This is a neuron in the reticular formation of the brainstem. It's been counterstained for a dye that labels the nuclei of surrounding cells, and here in the nucleus of the neuron. And the pink stuff labels the nissl stuff in the cell. And these little black dots are injection of mercury that was injected subcutaneous on the back. Heavy metals also transport into the brain. That's why this is one of my favorite neurons, because you can see where by staining it with a particular procedure, you can see where the neurons are that are labeled with things like environmental toxins. Okay, I have more, but I think I'm going to stop there. If you want more, I can talk about them if you want.
Great, thank you, George. Are there any questions, comments?
This is Louis Morel again. Sir, a few minutes ago, you talked about amphetamine attaching to some receptors, and you showed a graph that showed craving as a function of binding of a protein or to a protein.
Right.
I'm curious to know how to interpret the values on the binding protein axis, the x-axis, because some of those values are negative. Can you explain that?
Yeah, because they're looking at the values relative to a baseline. So in fact, there's always something binding in the brain by displacing the receptors. You can have, if we're talking about the graph means that you're going from getting a high to something. Let me go, I have to go back and look.
Sure.
I anticipated that question, but I don't remember the answer. Let's go back to see where we were here. Probably, nope, not there. Oh, they all, you know what? None of these have names. Let me think for a second. Let me go back a second here. Nope, that's not the one. Oh, yeah.
That's the one.
Oh, we're talking about the binding becoming negative is because there's a baseline amount of binding that's down in the middle. And as they start to go low baseline by displacing, normally they're displacing the drug that gives you the craving, but you can also displace the normal dopamine that's there, which can increase your craving and reduce the amount of binding of the amphetamine. I think I've got it right, but I can get to the paper for you and clear it up. I should have thought about that a little closer. I took it for granted of a normal baseline, but I forgot that negative might be confusing to people.
No, and that makes sense. Hi, this is Ellie Vance. It makes sense, doesn't it? Because the cravings, it kind of raises that level, the need for the dopamine to be, how would you say? It does something with the normal level for stimulation, doesn't it?
So one of the things that was known for a long time, and even in studies, when I was looking at behavioral studies, when you interrupt some axon passages, this was known in the peripheral nervous system. It was also known in the brainstem. If you take away an input like the dopamine input to the ventral striatum or dorsal striatum, animals recover. And at least one reason they thought they recover, and one reason why Parkinson's patients don't show a lot of symptoms in the beginning is they thought that as they have lesser and less input, the receptors on the cells multiply to make use of the little input that comes in and still get the same behavioral output. So if you have a neuron, you take away one input, but you still have one left, well, it duplicates the receptors, and this goes over and innervates more receptors to try and maintain homeostasis in behavior.
Right. Yeah, there you go.
So this upregulation of behavior is something that can measure biochemically and in neurochemistry. You can measure it for dopamine, you can measure it for other compounds called norepinephrine, and it's thought to be important in recovery of function or maintaining function, especially in something like Parkinson's disease, where the loss of dopamine to the striatum is thought to precede the loss of the really overt severe symptoms that these patients experience. And one idea is that they're multiplying their receptors and trying to keep up until they can't keep up anymore. And
Right.
there's a way you can test this by giving a pre-Parkinson's patient that you suspect a dopamine blocker, and then they crash. But they don't do this because when they crash, they're worried about the fact, and I think in rats that they don't come back from the crash. So you have to be a little worried about giving a test for upregulation in a human pathological case. But you can deplete dopamine in rats and show that kind of phenomenon.
Most interesting. Yeah, my background is more in the study of behavior of people, and then trying to connect that with a much more basic level of understanding for the chemistry behind it.
Mm-hmm. Well, there's some of the articles
Thank you.
I can forward to. I didn't put all the references in the readings because I hadn't finished all the rest of it. To be honest, I was working out some ideas of which direction it might go. So I actually have two more directions to go in. I can show you, but the readings themselves, this paper by Volko, some of these are in places where they can be downloaded for free and you can read. Some of them are a little technical and hard to read, but if you start to understand the anatomy and the background and have a little bit of the terminology, then you might be able to read the articles and get what I'm talking about. Some of the things are very basic, the idea between everybody's heard about Pavlov's dogs, I think.
Oh, sure.
And the idea that you can ring a bell and prevent food and the animal starts to salivate.
Mhm.
Eventually, when you ring a bell, they start to salivate. Well,
Mhm.
if you have somebody like with a patient who's drug addicted, they know that if they come into a certain location and area, the context is there that Jim the Eskimo's coming with the drugs, you start to get the cravings because you're in a place where that drug's going to be delivered. And
Right.
for rats, you can set it up so you can ring a bell and give them the drug, ring a bell and give them the drug, and they'll come and go to a certain spot or press a lever. But you can also extinguish them, and this is a good test for drug addiction. You can ring a bell, give them no drug, and keep ringing the bell until they ignore it. They're not going to respond to the bell at all because they know no drug is coming. But if you give them a shot of the drug, they'll go right over and start pressing the lever. It's called a drug reinstatement of responding.
There you go. Yeah.
And that's
Thank you,
blocked,
George.
and that's blocked by blocking the pathways from the posterior basolateral to the extended amygdala. If you do it another way and you drive them through extinction and you, instead of giving them a drop of drug, you ring the bell and give one shot of the drug, they'll go start responding again because the drug itself is a stimulus, an internal stimulus to start responding. Say, the drug's back, go back to work. And this is why part of the two parts that make drug addiction really difficult. The third part has to do with the things in the temporal lobe related to depression and everything else that goes along with crashing these receptors and their normal output pathways.
Yep.
Okay.
Very good. Thank you, George.
Great. Thank you.
One last thing I'm going to recommend before I wrap up, and that is, I think in here I have this. This paper is free online and is almost the best paper I've read in physiology and neuroanatomy in 50 years. And it's in Cell Research. If you look on PubMed and you Google, or you can just Google on PubMed. If you go something called PubMed and Google hand W and this guy, Oraho, E. Oh, I said turn off. Can you hear me, Kim?
Yes.
Yep.
Google this paper, you can download it for free. He shows how the gut stimulation in pathways from the gut are actually rewarding. You can stimulate the ganglia in the throat of a mouse, and they'll press a lever or go to a space to receive that brain stimulation. Not only that, that part of the ganglia projects to the substantia nigra, which then projects to the striatum. And you can measure that because when you stimulate the ganglion here, you can measure dopamine release in the striatum. So that part of the peripheral nervous system is part of the reward mechanism. And the signal for reward is nutrients in the upper gastrointestinal tract. And this paper follows these pathways all the way from the gut through the substantia nigra, through a relay in the midbrain, in the brainstem, to substantia nigra and the dopamine, all in one paper. It's terrific. Go look it up. It's right down hand. It's got to be a paper, and it uses every modern technique in neuroanatomy, including genetic labeling. So it's my hooray for brain research recommendation this day. Okay.
Wonderful.
Thank you, George. Appreciate it.
Wow.
Anybody else have any comments?
Yeah. Somebody commented that it's just fascinating and complex. And I think we would all agree that it is both complex and fascinating. We appreciate your presentation today.
Thanks.
Thank you.
I'm happy to do it.
Yes, thank you, George.
It's like asking someone if he has any slides of his kids.
So thank you. I know that a good number of people have dropped off just because we ran a little bit over, but we do appreciate you taking a little bit of extra time to wrap everything and especially share that last paper with the class. So with that, I will go ahead and close the virtual classroom if no one has anything else to say.
Nope. It's fascinating.
Yeah, we appreciate it very much.
I hope you enjoy it and keep looking for it. These things turn up in the newspapers. They're getting to be so fabulous now, and it's getting to be news. Enjoy.
Good. Okay, goodbye, everyone. Have an awesome day.